All right. So you should have control of it.
Okay, so
And
it should say
yep,
that work?
yep. So your PowerPoint is up there and you are good to go. So thank you.
Oh, great. And Sheila, I can see questions, so if you type them in, I can see them as they go, apparently.
Oh, good. Okay, perfect.
All right. So good morning and or afternoon wherever you are. Thank you for listening to my story. I'm going to tell you a little bit about the work that my lab is doing using induced pluripotent stem cells to study Angelman syndrome. I'll explain a little bit more about what those cells are in a second, so don't freak out yet. I'm at the Department of Genetics and Genome Sciences at the University of Connecticut Health Center in Farmington, Connecticut. So any of you folks who are local, feel free to send me an email and drop by, and you can see what we do in the lab in person if you like. So you probably know this already, that there are different types of cells in your body. I'll talk a little bit today about fibroblasts, which are one type of skin cell. I'll definitely talk about neurons, which are a very important cell in the brain. But I'll also talk about stem cells. And there are two major types of stem cells, adult stem cells and embryonic stem cells. And I'm going to spend most of the time talking about embryonic type stem cells. Adult stem cells can only make the cells of one organ. So for instance, blood stem cells only make blood. Neural stem cells only make brain, skin stem cells only make skin, so on and so forth. But embryonic stem cells are different in that they can make any tissue type of the human body. We all have adult stem cells in our body at any given time, but we do not have embryonic stem cells, except for perhaps those of you who are pregnant. You might have some embryonic stem cells with you, but the rest of us do not. And so embryonic stem cells are really cells that exist only for a small period of time in an early embryo. So what are these induced pluripotent stem cells then that I speak of? And I abbreviate this IPSCs or IPSCs. So I just spent a little bit of time telling you that human embryonic stem cells can become any tissue type in the human body. But the other thing that's cool about these cells is that they can be grown in the lab indefinitely. And where human embryonic stem cells come from is that they are generated from discarded human in vitro fertilization embryos. So when couples try to conceive a child through IVF, they typically would donate their leftover embryos to stem cell research, and that's how we have human embryonic stem cells in the lab. So to be clear, these cells are usually created originally for reproductive purposes, but before they're discarded, families might decide to contribute them to science instead. And so they have some ethical implications in that, some religions don't believe in destroying human life regardless of where it comes from. And so there's always an ethical problem with human embryonic stem cells. And remember, they were always created for reproductive purposes. So there's a limit to the number and types of embryos that are available for embryonic stem cell research. The cells that I use, however, are different. They're functionally equivalent to human embryonic stem cells. They can be grown in the lab indefinitely, and they can become any tissue type in the human body. But they weren't generated from embryos. They're actually generated by turning on stem cell genes in skin or blood cells. And so we can take skin samples or blood samples from patients and turn the cells in those samples into these stem cells that do everything that a human embryonic stem cell can do. And so those are our induced pluripotent stem cells. So again, what we do is we would take samples, skin samples or blood samples from Angelman syndrome patients. So far, we've only done this with deletion positive patients and patients with mutations in UBE3A. We can change those cells into stem cells, and the stem cells look different, as you can see on the picture at the top here. And then, so we make these iPS cells by expressing the stem cell genes in the skin cells or blood cells. And then the stem cell genes program the cell to become a stem cell. Okay, and from that point on, they're stem cells and no longer skin cells or blood cells. So every time we've made these iPS cells, so we call them iPS cell lines, each line is verified as a stem cell, and we re-diagnose them with Angelman syndrome to make sure that we have the cell that we think we do. And so I'm showing you an example from a paper in our lab where these are 3 different stem cell lines that we produced from an Angelman syndrome patient with a large deletion. The green spots show staining with a specific marker that just tells us that they really are stem cells. And so, and the point of this is to know exactly what we're dealing with. We really do have the stem cells that we say we have. At the bottom of this slide, the other thing we do with these cells is we always perform a karyotype, and this is where we look at the chromosomes, and we try to make sure at the chromosomal level that they have the Angelman syndrome deletion. So I personally couldn't tell you whether or not these karyotypes tell us that they have Angelman syndrome, but the aficionados, the people who do this professionally, can. And so they looked at these chromosomes and told us that they had a deletion. And we also performed something called DNA FISH, to show that they had the deletion that led to Angelman syndrome in this case. And I'll explain to you that we also do a methylation test here in a second. So we've derived iPS cells from patients with Angelman syndrome, and I told you before that we've only done it from deletion positive patients and patients with UBE3A mutations. But this is the list of iPS cells that we've derived so far from Angelman syndrome patients. Interestingly, we have the UBE3A mutation patients. One of them has a 2 base pair deletion. And then another patient has a 1 base pair missense mutation, which means that it's just 1 base pair that's changed and not missing. What's really neat about this patient is the family had 2 boys that were affected with Angelman syndrome, and they both had this mutation. The mutation was inherited from their mother, so we sampled the mother and made iPS cells from the mother, and then she inherited the mutation from her father, and so we also sampled him and made iPS cells from the grandfather of the affected individual. And so we have a nice family here with Angelman syndrome. And going forward, and of course we had to make iPS cells from typically developing individuals, and we do that so we have something to compare the Angelman syndrome lines to. And in terms of making more stem cells, right now we have 2 major goals. We'd like to recruit individuals with paternal uniparental disomy, and individuals with imprinting defects, because those are not represented in our stem cell lines yet. And there's a reason that I hope I'll remember to tell you about later, why specifically we're interested in these people. And we're also, we'd also like to recruit individuals with rare mutations in UBE3A that may have an intermediate phenotype, and so for instance phrase speech, because we think that this might help us understand how UBE3A contributes to different pieces of the Angelman syndrome features that your kids have. This is a simple map of the chromosome 15q11 to q13 region, the region that's missing in your kids. And I've drawn on here breakpoint 1 and breakpoint 2 and breakpoint 3. Many of you may know, especially those of you with deletion positive kids might know whether your child is a type 1 or type 2 deletion. And importantly, here is the UBE3A gene. It's shown in red on mom's copy. So I labeled Matt, and Pat means paternal, Matt means maternal. UBE3A is only expressed from mom's copy in neurons, and that's why it's in red. The other genes that are shown in blue are expressed only from dad's copy. And that's true of all cell types. I drew this map because I wanted to point out that the region is regulated by an imprinting center, and this has a methylation imprint. It's unmethylated on dad's copy, and it's methylated on mom's copy. In this methylation, many of you, your kids have had a methylation test to determine whether or not they had Angelman syndrome. And this is important. This region, this imprinting center controls the gene expression over the entire region. And so one of the things we had to look at in our stem cell lines was to look at the methylation imprint here to make sure that the cells retain that through the reprogramming process. So when you turn a skin cell into a stem cell, we know that it erases DNA methylation sometimes, and we needed to make sure that this imprint didn't get erased. And so we did the methylation test in our IPS cells to make sure that they're like the Angelman patient samples. So this is the exact same test. If your kid was being diagnosed with Angelman syndrome in Connecticut, this very test may have been run on your child, where we can look at whether the child has a maternal allele or a paternal allele at this imprinting center. And so panel A shows the maternal allele and panel B shows the paternal allele. And then we see normal or a typical developing child that would have both a maternal and a paternal allele. And then our two Angelman stem cell samples that show only the paternal allele, and then for control a Prader-Willi sample that shows only the maternal allele. And we could quantify this methylation and show that in normal IPS cells, we see about 50% methylation because they have one maternal allele and one paternal allele. And then in our Angelman sample, we see no methylation, less than 1% methylation, indicating that they only have the paternal allele. And so this told us that the IPS cells are retaining the methylation imprint and that they would be diagnosed as Angelman syndrome. So we made the stem cells, we converted them from the skin cells or blood cells, and then we need to convert the stem cells into neurons because, as many of you know, the neurons are the brain cells that are affected in your kids. And I need to point out here that we don't make the stem cells or the neurons for replacement therapy. There's a lot of people assume that because we're doing stem cell research that the point is that we want to put these cells, the stem cells or the neurons, back into kids, and we really don't. We don't think that that would be useful for Angelman syndrome. Your kids have a brain that looks pretty normal. The neurons seem to be in the right place, and they seem to largely be doing the right things. And so, if we tried to put neurons back there, it would probably be disastrous. So we think that there are a lot of other really cool things, though, that you can do with these cells, and I'll tell you some of those things that we're trying to do. So we had to do one more thing before we could move on with the models, and we needed to ask whether or not the neurons have Angelman syndrome. So UBE3A is interesting in that it's, in most tissues, it's made from both parental alleles. So it's made from dad's copy and it's made from mom's copy. And so in our stem cells, there's actually UBE3A made from dad's copy. But in neurons, an antisense transcript called UBE3A antisense or UBE3A ATS is made specifically in neurons, and it silences dad's copy of UBE3A. And we needed to make sure that this happened when we turned our stem cells into neurons. So we asked that question, and to ask the question, we looked at 3 specific genes in this region to see whether or not they made, the neurons made the antisense transcript. So in purple, we looked at SNORD116. That should be made in all cell types in humans. And so I means IPS cells, P means neural progenitors. These are actually neural stem cells, an adult stem cell that I was telling you about earlier. And then N means neuron, and specifically old neurons that are fully functional. So SNORD116 is made in all of these cell types. SNORD115, which is a little more neuron specific, and then UBE3A antisense, which is the part of the gene that overlaps UBE3A and really silences UBE3A. we found that they're made in either neural stem cells and neurons or just neurons alone. And so this tells us that the UBE3A antisense is made only in neurons, like it is during human development, and actually during mouse development as well. And we also looked at UBE3A protein levels in our IPS cells and in our neurons. So I is again IPS cell, N is neuron, to make sure that UBE3A protein went away in our Angelman samples. And so this told us that since UBE3A protein is going away in our Angelman syndrome neurons, that our Angelman syndrome IPS cells are good models for Angelman syndrome. And like your kids, they don't have any UBE3A in their brain. So to summarize what I've told you so far, skin cells or blood cells from children with Angelman syndrome have been turned into stem cells and are called IPS cells or IPSCs. And we've used the methylation test to make sure that the cells are like cells in Angelman patients. And we've shown that the IPS cells make UBE3A antisense when they become neurons. And that the IPS cells silence UBE3A when they become neurons, just like Angelman syndrome neurons. So then, now that we know we have Angelman syndrome neurons, and so again, these are live human neurons with Angelman syndrome, what can we do with them that's useful? And so three things come to mind, and I told you earlier that we can't use these, we don't really want to use these for replacement therapy. So one thing we can use them for is to learn how UBE3A becomes imprinted in neurons. How does dad's copy of UBE3A get shut down? The second thing we can do is learn what is wrong with the Angelman syndrome neurons. And the third thing that I'll tell you about is how we can use them to identify and test potential therapeutics for Angelman syndrome. So first, one of the major ongoing stories in my lab is to learn how UBE3A becomes imprinted. So we're trying to ask the question is, why is the antisense transcript different in stem cells and neurons? In stem cells, part of the antisense is made, but it stops. It stops at this gene called IPW. And in neurons, it goes further and makes UBE3A antisense and silences dad's copy. And so the way we kind of think about this is that there's a stop sign in most cell types. And all of the cell types that aren't neurons, there's a stop sign here that stops the antisense transcript from silencing dad's copy of UBE3A. And we want to understand this because we think that if we can figure out a way to maintain or replace the stop sign in neurons, that might be a potential therapeutic approach for Angelman syndrome. It's important to do this in human cells because mice regulate this a little bit differently than humans do. And so the fact that humans make part of the antisense transcript in non-neurons gives us another potential way to unsilence dad's copy of UBE3A. And we think that a specific protein called CTCF may be the stop sign. And I won't bore you with the details of how why we started looking at CTCF, but you know, to make a long and tough story short, we found that by comparing by looking at the maternal allele versus the paternal allele, so we did this by comparing Angelman versus Prader-Willi syndrome IPS cells, we saw that this protein CTCF preferentially binds dad's allele, where the antisense transcript is made. And when we compare IPS cells to neurons, we found that CTCF goes away in the neurons. And this is consistent with it being a stop sign that has to go away in neurons when the antisense is fully made. And this isn't true for all CTCF sites. When we look at where CTCF binds at a different piece of DNA, and I should have said up front that we look at how this protein binds DNA, that's the assay that we look at here. If we look at a different place where CTCF binds, it doesn't look like that. It doesn't bind the paternal allele preferentially, and it doesn't go away in neurons. And so we really think that this property is specific to the stop sign that we're looking at. And this is the work of a graduate student in my lab, Jack Shao. So what we think is happening here is we think that CTCF is the stop sign, and what it does is it binds and it kind of ties a knot, making two loops out of this genomic region in stem cells and in non-neurons. And in neurons, when CTCF goes away, it makes one big loop, and that antisense transcript can then go through IPW and make the antisense transcript, and that's how it silences UBE3A. So that's our model. Recently, we got a little more fuel that our model might be right, because an individual was reported in the literature that has, well, several individuals were reported where they have the stop sign deleted. These individuals have Prader-Willi syndrome, so their deletions are on dad's copy of the chromosome. And there were at least 3 different individuals that were reported here that are missing the stop sign. And they all make the antisense transcript in non-neurons. So their stem cells, their skin cells, their blood cells make UBE3A antisense. And we showed in a different paper that when the antisense is made, it silences dad's copy, even in non-neurons. However, there was one patient that was recently reported that isn't missing the stop sign. They have a very similar deletion as these other patients. They also have Prader-Willi syndrome, but the stop sign is still there, and that patient still doesn't have any antisense transcript. And so we think in those patients, UBE3A is not imprinted in their non-neurons. And so this tells us that the stop sign is probably where we think it is, and that's right where those CTCF sites are binding. So what we're doing now is we're trying to remove the stop sign. So we can use DNA scissors called CRISPRs, and we can cut and remove the stop sign. And we want to ask whether or not the antisense transcript is made in the stem cells. So again, the non-neurons, is the antisense transcript made when we remove the stop sign. And that would help us prove that the stop sign is important for stopping the antisense from being made. The other thing we're trying to do is to try to scramble the letters so that it doesn't say stop anymore. And so CTCF binds to a particular DNA sequence. And so by scrambling that sequence, we think that, and again, it uses these CRISPR DNA scissors to do the trick. By scrambling the sequence, we think that it should be able to remove the stop sign, or the stop sign won't work anymore. And then we want to ask whether or not those cells make the antisense transcript and silence dad's copy of UBE3A. So that's how we're asking that question. Once we get those questions answered, the next logical step is to ask, how exactly does the antisense silence UBE3A, and are there places there that we could modulate to try to develop potential therapeutics? So next I'll tell you about what we're doing to learn what's wrong with Angelman syndrome neurons. And most of this is not work done in my lab, but I've given my stem cell models to many different groups, including a couple of different pharmaceutical companies, so that they can also study what's wrong with Angelman syndrome neurons. So one of the things that we can do is we can look at the Angelman neurons to see if they look different. On your left here is some pretty pictures of neurons that are stained with, in green, an antibody or a protein marker that tells us that we're looking at neurons. And then in red, little spots where one neuron can talk to another neuron. And we can look to see whether Angelman syndrome neurons look the same as typical neurons. And we can look at the spots, and we can look at the neurons themselves to see if they look similar. The other thing we can do is, so the spots are actually localized on these little pieces of neuron called dendritic spines. And Scott Dindo's group back in 2008 showed that Angelman, in looking at Angelman mice compared to typically developing mice, that the Angelman mice had fewer of these little dendritic spines, and that they didn't have the same shape as typically developing mice. And so we've been able to, in collaboration with Dr. Les Lowe's lab, look at the dendritic spines on human stem cell derived neurons. This isn't actually from an Angelman patient. This is actually from a patient with too much UBE3A instead. And so far it seems like neurons with too much UBE3A actually have too many spines. And so they're now looking at Angelman syndrome neurons to see whether or not they have too few spines as we might expect. And again, this is done in live human neurons, and so we're really asking of the human neurons whether they show the same features as the mouse. We are also collaborating with another neuroscientist, Dr. Eric Levine. He previously had received funding from the Angelman Syndrome Foundation to do this, to study the basic functional properties of Angelman syndrome neurons. And so he's measuring the ability of these neurons to send and receive signals, much like some of the work that's already been done in mouse, but again, you have to ask the same question of human neurons. So on the left, these are traces of individual neurons showing their ability to send signals. The top trace here is an immature neuron, and then the bottom trace is a trace that's indicative of a very mature neuron. And so again, these are the electrical signals that neurons are sending. He can also look at the signals that the neurons are receiving. I left the big words up here to talk about these neuron properties for any of you who are really into neurons. I'm a terrible neuroscientist, so I'm not going to bother explaining, giving you my poor explanation of them. But how Eric does this is he patches into individual neurons and sticks an electrode in those neurons to measure the electrical activity in these neurons. And through doing this, he's been able to ask whether or not the Angelman syndrome neurons are delayed in their development. And what you see here is a graph showing something called resting membrane potential of neurons. And in general, this is a nice measure of how mature neurons are. The neurons that we make in a dish from our stem cells really are equivalent to fetal neurons. And as you can see, they're developed over about 20 weeks in culture, which is really, if you think about your pregnancies, they were only about halfway through your pregnancy. And so we think that these are still fetal neurons. And what you expect to see that is as these neurons develop, they get lower and lower resting membrane potentials. So these are negative numbers. And the lower resting membrane potential means that they're more mature. And what Eric was able to see was that in control neurons, or typically developing neurons, that happens. Over development, they in general have lower resting membrane potentials, but the Angelman neurons seem to have higher resting membrane potentials, and they're not seeming to go down with age as we might expect. So some of the early indications are that, that the neurons may be delayed in their development even at the fetal stages. Eric is wrapping up these studies, and they should be submitted for publication soon. They were actually submitted once and rejected, so hopefully they'll get out this time in a different journal. My lab is also developing something they're called cerebral organoid cultures. We also call them brain bits. So the neurons that we typically grow are stuck to a piece of plastic in a dish. Not at all like how a brain develops, but we've learned ways to let these neurons develop in three-dimensional structures that look a lot like small little brains. So these are tiny. They're only about 3 to 4 millimeters in size, so they're nowhere near the size of a regular human brain. But they developmentally seem to behave like a human brain. So in red you can see a neural stem cell compartment. So these are the neural stem cells. You have cells like this in your brain as we speak. And then from those stem cells, they develop out into layers. And the green stain shows the more mature neurons that are developing into the proper brain layers. And so we've developed this model so that we can ask how the Angelman syndrome brain develops using tissue that's a little more similar to the human brain. And so in thinking about Eric's finding that perhaps Angelman neurons have delayed development, we want to know, can we see this in a mini brain with Angelman syndrome? So the third thing that we can do with our Angelman syndrome neurons is, and probably the most important thing that we can do with these neurons, is to use them to identify and test potential therapeutics. And so we've already worked with Ben Philpott's group to test topotecan to ask whether or not it also works in human neurons. And I'm showing you data here from our lab where we've treated our Angelman syndrome stem cell derived neurons with topotecan at increasing concentrations. In blue, the blue bars show UBE3A antisense, and the red bars show UBE3A mRNA. So this tells us whether or not these genes are made. Topotecan works. Ben had found that topotecan works by decreasing the antisense transcript and increasing UBE3A, and we could show that that happens when we treat human neurons with topotecan. But we did learn that in humans it takes a longer treatment and a higher dose than what's used in mouse. And so it's a nice thing to be aware of as studies move forward to investigate whether or not this is going to be a good drug or something like it is going to be a good drug for Angelman syndrome. We've also worked with Ben's lab and Mark Zilka's lab to see whether the drug mechanisms work the same in mouse and humans. So again, topotecan seems to work by reducing the amount of genes that are really, really long. And so we were able to use our human neurons and show that when we treated them with topotecan, really, really long genes were expressed at lower levels compared to shorter genes up here. And so it was nice to be able to show that the mechanisms are the same in mouse and human. And so in that case, it means that the mouse is a very good model to look at how topotecan works. Similarly, we can use our models to look at antisense oligos. And so this is the paper that was recently reported by Dr. Art Baudet's group along with ISIS Pharmaceuticals. And they showed that in mouse, they could use antisense oligonucleotides to, again, reduce the antisense transcript level, and that restored paternal UBE3A expression. It's actually particularly it's particularly important in this case to test these antisense oligos in human cells because these are DNA drugs, and the DNA sequence is different between mouse and human. So you actually have to go to human cells to develop the specific antisense oligos that would work as a drug in Angelman syndrome patients. So my lab is using the iPS cell derived neurons as well as Prader-Willi cells to identify the human antisense oligos that might work, and we've shared these cells with ISIS Pharmaceuticals so that they can they're doing the large-scale screening, and then they ask us for help every now and then to test the antisense oligos in our lab. So so that work is ongoing. You know, I'm not allowed to show you data, but it does seem to be promising in human cells, and rest assured that ISIS is still working on that problem. So with the therapeutics, the potential therapeutics that are coming up, we kind of wondered whether we can establish a system for testing potential therapies in the human system. We don't have mice that have there are Angelman syndrome mice, of course, but the mice have a limited number of behaviors that aren't exactly like the human behaviors, and in our human cell culture models we have no behaviors at all. So, but we wanted to figure out whether or not we can develop a system where we can really test whether or not therapies work well in humans. At least in the cell culture system that we're using. And so what we've done is we've developed IPS cells that are genetically identical to one another except for a UBE3A mutation. So we took one of our Angelman syndrome IPS cell lines that had a mutation in UBE3A, and again we use these DNA scissors to replace the changed base pair and change it back to the DNA that it should have. So the isogenic control line down here has a normal UBE3A copy and does not have Angelman syndrome, but it's genetically identical except for this mutation in UBE3A to its Angelman syndrome counterpart. And so think of this as having identical twins, one of which has Angelman syndrome and one doesn't. So we can compare these and know that any difference that we see between the Angelman syndrome cell line and the typically developing control cell line has to be due to Angelman syndrome and not due to the inherent differences between individuals. So we've made this cell line in the lab. It turns out we can do this really efficiently, and it works really super well. So we've decided we're going to correct the second UBE3A mutation line. So we have 2 examples of cell lines that we can use. So what we'd like, in addition to those cell lines, there's another way that we can make this these identical twins with and without Angelman syndrome. And don't get me wrong, I don't mean to make people. We mean to make cell culture models of neurons that we can study. The alternate strategy is to take a mosaic patient where some cells have Angelman syndrome and some cells do not, to take a blood sample from a mosaic patient and isolate stem cells with Angelman syndrome and those without. So those would also be identical twins with and without Angelman syndrome that then we could use to test potential therapies. And these, I mentioned earlier that we're really interested in imprinting defect individuals. The mosaic patients are most likely to be imprinting defect individuals, and so that's one of the reasons we're looking for those patients. And so once we have now that we have these identical twin cell lines with and without Angelman syndrome, what we'd like to do is make them into neurons and compare gene expression across the whole genome to identify genes that are higher or lower in neurons with Angelman syndrome. And so I kind of envision this as if you can imagine Angelman syndrome versus typically developing neurons being on a spectrum. If we can treat our Angelman syndrome neurons with a potential therapy like an antisense oligo or topotecan, we can figure out where on the spectrum treatment lets these cells land. Can we get them all the way so that they look like typically developing neurons, or are they going to fall somewhere in the spectrum between Angelman syndrome and typically developing neurons? And so by having the spectrum, we can understand what dose of the drug might work best and whether or not there's a window for optimum treatment. Of course, we can do this in the mouse, but we worry that it might be different in humans, and so we think it's worthwhile to go through and look at this in the human case. The other really cool thing that it does is it allows us to test different Angelman syndrome mutations to see whether they're full-blown Angelman syndrome or whether they're more similar to typically developing individuals. So for instance, I understand that there's a family with a mutation in one of the isoforms of UBE3A. So we could, for instance, make that same mutation in our stem cell lines and ask, are they closer to the typically developing cells, or are they closer to the Angelman cells? And the genes that are changed in those cells might be involved in a certain aspect of Angelman syndrome. So for instance, if those patients have better speech than the typical Angelman syndrome child, maybe those are the genes involved in speech development. And so that's the other thing that we can do with this cell culture model where we have identical twin cell lines, one with Angelman syndrome and one without. And ultimately, those individual genes that we identify that differ between Angelman syndrome and typically developing neurons, and especially those that we can correct with a potential therapeutic, they may be biomarkers to determine whether a therapy is working. And so what I'm showing you here is data that we have by comparing whole genome gene expression between Angelman neurons and typically developing neurons. In yellow are the genes that are have higher levels of expression in Angelman syndrome neurons than their typical counterparts, and those that are lower in Angelman syndrome versus their typical counterparts. So these genes that are different, maybe we can look in the blood or cerebral spinal fluid samples from Angelman patients or Angelman patients treated with potential therapeutics once clinical trials get underway to determine whether or not the drug is doing its job in patients. And what's the reason we think that this will be important is that your kids have difficulty in development and in learning and memory and in speech but those are all things that take a long time to acquire. So for instance, even in a typically developing child, language development takes several years. And so if we're able to restore UBE3A in an Angelman syndrome patient, it's still going to take a long time for those kids to learn how to speak, for instance, if the therapy works as we all hope it does. And so one thing that we can do is try to predict whether or not the drug is doing its job in restoring UBE3A expression. And we want to make sure we're able to do that so that drug companies don't prematurely stop clinical trials when a child that gets the drug all of a sudden doesn't just start speaking, which we don't really think that they would. So that's some of the reasons that we do what we do with the gene expression and looking for potential biomarkers. And I know biomarkers is a tricky word. Others are using biomarkers in different ways. And the biomarkers that I speak of are specifically gene expression levels that we're looking at. And so the story with the genetically identical stem cell lines and then looking at gene expression between the neurons derived from those cell lines, that's the work that's been recently funded by the Angelman Syndrome Foundation, and these studies are well underway, and we're really excited about them. So to summarize, we use IPS cells to study Angelman syndrome in live human neurons. And we think that the Angelman syndrome mouse is a great tool, but we recognize that there are some things that are different between mice and humans. And so we hope that this human stem cell model is really a complementary model that allows us to really confirm findings in the Angelman mice and to learn a few things that are new and might be human specific. We want to know what human neurons with Angelman syndrome look like and how they behave. And so we do that in our lab, but largely a lot of that work is done by collaborating with other people. We're happy to share our stem cell lines. In fact, they're commercially available to any scientist who would like to purchase them. We've purposely made them really inexpensive to get, and if money is an object, I'll send them for free. So we feel passionate about sharing our models. We can use our human Angelman syndrome neurons to test drugs or identify new drugs. And we think that this is actually going to be most useful in combination with mouse studies. It's a little faster and easier to do certain studies using mouse neurons or using mice. And so we'll fill in the gap as we may with the human neurons. And the reason to do this is it might help us know whether the drugs will work actually in your angels and just make us feel better going forward with clinical trials. So with that, I'll point out the individuals in Connecticut who are working on Angelman syndrome. We were all at the Angelman syndrome walk last weekend, and I'll point out the people. This isn't all my lab. My lab consists of 3 PhD students, Ivy Chen, Jack Xiao, and Carissa Sirois. 1 MD PhD student, Ali Getchin, she just joined the lab recently, and a postdoc, Noelle Germain. The other people are from some of them are from Mark Lalonde's lab. They work on Prader-Willi syndrome, but it's important to study Prader-Willi syndrome because there's some genetic overlap here. And then a couple of folks from Eric Levine's lab, Tawana and Kia. And then Chris Stoddard is an excellent technician who makes some of the DNA scissors and such that we need to make the twin cell lines that I told you about. And I collaborate, I mentioned Eric and Les. I also collaborate with a genomicist, Brent Gravely, here at UConn, as well as Ben Philpott and Larry Ryder. I've collaborated with others. I need to thank the families who donated samples and who will donate samples in the future. And then my funding sources, the NIH, the Connecticut Department of Public Health, Raymond and Beverly Sackler Foundation, Fighting Angels Foundation, a young Angelman Syndrome Foundation group that's started up in Connecticut, and then of course the Angelman Syndrome Foundation, who I'm very grateful for their support. And with that, I'll take any questions.
Hey, Stormy, sorry about that. I couldn't get myself off mute there. Okay, so we had a couple questions.
Okay.
We had a couple questions come through. One of them is,
Okay.
can you define what a biomarker is and is not? So just kind of as a general point of information for the community.
Yeah, good point. So biomarkers mean different things to different people. And so it's a very good question, and you should always ask it. So in some cases, biomarkers mean, so for instance, one might call an EEG a biomarker because an EEG might tell, in a clinical trial, somebody could use an EEG to say whether or not a drug is working to fix the patient's seizures. And so that's one definition of a biomarker. And the definition of the biomarker that I'm using is gene expression. So we're just using it to see whether or not UBE3A levels and other genes that change when UBE3A levels change, whether those act like we think they should when we restore UBE3A levels. So biomarker simply means something that moves, that changes when you've corrected or treated something so that you can know whether or not your treatments worked. I hope that makes sense.
Yes. So another one, and actually they kind of go together with another one that we just got. Let me find it. Somebody wants to know if you will be recruiting kids with ICD and UPD for donating samples. Or are you looking for participants to grow the stem cell lines?
Yeah, so let's see. So we are looking for, we will be recruiting ID and UPD patients soon. We're waiting for our IRB. We had to modify our IRB because we're no longer taking skin samples. We're only taking blood samples. My bar was, would I take my 2-year-old or my 4-year-old to get their skin sample taken, and I kept telling myself no way. And so we can now do it from a small sample of blood. And so we're just waiting for our IRB approval to do that, and then we will be taking those patients. And I'd really like to find mosaic individuals with kids with mosaic, that are mosaic for Angelman syndrome, because it allows us to generate, again, the twin lines that tell us a little bit more. And the other question was, were they both recruitment questions, or?
Yeah, they were both along those lines. Yep.
Okay.
And then we do have some more. Let me take a look. We just got a couple more in. Have you tested if artificial transcription factors made at Siegel's lab activate the paternal UBE3A?
We haven't yet. We have the tools to do that. We have the tools to do that a little bit, and so it's something we've kicked around. And we haven't done it because my hypothesis was it probably wouldn't work, but that's not a good reason not to do it, and so we'll probably try that because we can. But it's a really good idea.
Okay. Then we had another question back to relating to the biomarkers. So are biomarkers objective and measurable?
So that's why we've chosen to look at whole genome gene expression, because it's very measurable, and by looking at enough samples, we can know how much variability there is, so we can know whether or not in response to treatment the change is bigger than the variability, the background, the noise, so whether the signal's better than the noise. So that's one thing that's beautiful about looking at gene expression, is it's quantitative. And it can be done in a very high throughput manner.
All right, and then we have, it looks like, one final question. Let me just make sure. If anybody has any that they want to get in, go ahead and send them in now. And I'm sorry, I'll probably butcher the name, but other than topotecan and the ACOs, are there other compounds or drugs you have tested using IPSCs, or can't you discuss?
Yeah, no, so we haven't tested anything else. We haven't tested anything other than topotecan and the antisense oligos. I have been contacted by a couple of different pharmaceutical companies that had different, so one of them shared with me their approach, and in fact it was a company who was they work in gene therapy a little bit and modulating. So they have a technology that allows them to precisely modulate UBE3A levels when it's provided like in a gene therapy type context. And so they weren't necessarily asking me to help them test compounds. They more or less were discussing whether or not the approach would work and whether or not the stem cell neurons could be used to test their particular approach. So, there are other companies interested, and I think that there are people doing work, other companies doing work, but we haven't tested anything else ourselves.
All right. Well, it looks like that's all the questions that came through. If anybody else has any questions that they think of at a later time, you can go ahead and send them to info@angelman.org, and we will try to get those answered for you. Want to thank Dr. Chamberlain for joining us today, and we greatly appreciate you putting on this webinar for us. And again, everybody, it will be recorded, so you guys can watch it probably later today if we can get it up. Other than that, have a good Memorial Day weekend.
Thank you. Thank you all for your questions.
Thanks,
Have a good weekend.
Bye.
Bye.